Hello everybody, so today I'm going to be discussing a paper from the Nutritional Journal of Biochemistry. This paper is on perinatal exposure of rats to a maternal diet with varying protein quality and quantity and how it affects the female offspring. So several experimental data has shown that nutrition is a perinatal programming factor that regulates major tissue and organ development. So this has the chance of offspring developing metabolic issues later on, such as diabetes type 2 and just being overweight in general, which can cause lots of other health problems. So in utero, the exposure to an excess or restriction to energy or macronutrients during gestation can program food intake control issues like hyperphagia, which is excessive eating. So the authors of this article conducted a study in rats to evaluate the effects of protein quality and quantity in the maternal diet during gestation and lactation. So this study focused specifically on the weight and adiposity of adult female offspring. So the presented study in rats used two different levels of protein, a normal protein at 19% and a high protein at 47%, along with three different types of proteins, cow's milk, peas, and turkey. So sufficient levels of both protein and amino acids during gestation and lactation are nutrition programming factors for optimal health in the offspring. So when comparing the animal protein to plant-based protein, protein in the plant-based diet was associated with poorer digestibility and lower content in the essential amino acids methionine, lysine, and tryptophan. So this study was conducted using Western rats. They were maintained under controlled conditions of 22 degrees Celsius. They had a 12-hour light cycle beginning at 8 a.m. And a 12-hour dark cycle, and they had free access to food and water at all times. So specific characteristics of the diets included essential amino acids and fatty acid compositions. 6 groups of dams were fed a high-protein normal diet, isocaloric diet during gestation, and during lactation, all of the dams received a normal protein diet from postnatal day 28 until postnatal day 70. So female pups were exposed to a normal protein diet or dietary self-selection. All other pups were only exposed to dietary self-selection. The dietary self-selection design was a choice between 5 food cups, one containing high-protein milk, high-protein peas, high-protein turkey, carbohydrates, or lipids. So during gestation and lactation, the weight and food intakes were recorded daily from postnatal day 28 until postnatal day 70. And overnight, on postnatal day 70, an overnight fast and fasting blood sample was conducted on the rats through a tail vein puncture. Adult pups in this study were then anesthetized under isofluorescence and balanced with nitrous oxide, and then they were decapitated. So after they were decapitated, the body compositions were assessed and weighed. So they weighed the carcass, the organs, and the fat tissue, both subcutaneous and visceral. So researchers conducted several biochemical assays in this study to quantify the cellular processes and reactions. So a couple examples of this is the Luminex assay, which was used to measure the plasma leptin, the magnetic bead multiplex assay, which was used to measure the metabolic hormones. So results of this study did show that high protein diets during gestation resulted in higher adiposity in female offspring, and exposure to different qualities during gestation and lactation affected the weight gain of the pups from birth to adulthood and their overall final adiposity percentage. So during lactation and post-weaning periods, the body was lower in the pea and turkey groups versus the milk groups. The dietary self-selection groups increased their total energy and fat intakes compared to normal protein weaning groups. In high-protein gestational groups, the total adipose tissue increase was associated with higher fasting plasma leptin. So these results suggested that maternal protein sources did impact offspring body weight, and protein excess during gestation, not taking the source into account, increased the risks of higher adiposity development in female adult offspring. During gestation, essential amino acid intakes were higher in the protein groups and were much lower in pea and turkey groups. So daily methionine and cysteine, I think that's how you say it, intake was lower in pea groups compared to milk and turkey groups. So milk was the leading group, turkey the second, and pea the last one. So this study showed that the quality and quantity obviously does affect weight gain and adiposity in female offspring. After weaning, the milk offspring gained more weight, and the pea groups showed to recover growth better than the turkey groups. So results of this study did lead researchers to believe that the maternal methionine cysteine intake was involved in these results, because methyl donors like methionine target gene promoters in essential organs such as the liver and the placenta. So this causes DNA methyl changes. So despite the varying results in several studies about this topic, it's been made clear that both high and low protein diets through the excess or restriction of amino acids does impair the development of offspring, regardless of the quantity or the quality the quantity of increased development in adiposity female offspring. So it does result in female offspring being overweight and resulting in lots of just health issues in general, like diabetes type 2, which is mainly why I decided to discuss this paper, because a lot of pregnant mothers don't know how what they eat is like, and how it affects their babies later on in life, especially female offspring. And this study does show that having a high protein diet does result in overweight female offspring, making these female offspring deal with diabetes type 2 and other overweight issues by the age of 20. So educating pregnant women on proper diet is really important. And thank you.